Hello and welcome back to Multibiomics, and we are continuing with the series Computational Genomics in R. The book that we are following is by the same title, Computational Genomics in R, and it is written by Altuna Akalin. So we were in the topic plotting with base graphs, and I have already explained it to you that these are the default some plotting functions that is provided by R, and we can visualize our biological data or any kind of data with the help of some plots. So we have started with histogram in the previous tutorial, and today we'll look at the how to visualize the data and compare a dummy genomic data set. So why a dummy genomic data set? So in the previous video tutorial, we tried to, we just had a first look on histograms. So we use random numbers to generate the plot. But in real bioinformatics, we don't work with random numbers. We work with biological data. And here today, we generate a dummy data set and try to simulate all the real world problems and noise, that a gene expression data has. So in this session, I'm not going to visualize or teach you, how to plot a histogram. Um, so I'm going to show you how to visualize the biological signals through histogram. Okay, like tumor versus normal. What are the tumor versus normal differences? How to visualize, co-express gene, even technical batch effects. And instead of downloading a dataset, we will actually simulate this realistic genomic data. And the structure you see is a typical one as you see in any RNA-seq experiments. Obviously, the scale and the size of this data is very, very tiny as compared to a real-world dataset. But why I have chosen this approach is because people with not so powerful PCs or laptops or students, they can generate this dummy data set, and at least they'll come to know about what kind of structure they are dealing with when it comes to data, right? They can have a look on matrices and all those things, gene expression matrix. So by the end of this video, you won't just know how to plot a histogram, but you'll also understand how to read biology from distributions. And this data set is simulated a gene expression matrix. We are going to generate a gene expression matrix. So this is the code. This we have used to simulate the data. What we are doing over here is we are making a gene expression matrix. I'll definitely explain it to you. It is very important to know about your biological data set. Without it, you cannot, proceed with your work. If you don't understand your data, you cannot solve the problem. You cannot analyze it. So first you have to do ED, exploratory data analysis, and all those things. If you don't know about how to do all these things, just there's so many resources which you can search. Free resources are available, paid resources are available, right? So you can go and you can learn. Even I'll try to upload such topics very soon, as soon as I get done with this series. Um, so what is happening here? First, this is for reproducibility. We are setting seed at 42. So every time the computers they generate random number, it's not random in true sense. There's a starting point, okay, and when you set the seed, okay, that's known as seed. If you set a particular seed and you use that every time, if you use every time, the same set of random numbers are generated, and you'll get same analysis, same output every time. Maybe, if everything goes right, you'll get same output because the same numbers are generated. So I prefer 42. I do not know why. I don't know why do I prefer, but you can use any kind of numbers. There are two parameters. There'll be two parameters, and once we are generated that matrix, I'll show you. So there are 60 samples from 60 patients, and we are analyzing, 100 genes. And in some genes, there are, the sample is divided by two, and half of the sample is tumor, and half of the sample would be assigned as normal. So here you see we are having some labels. These are the metadata. You have the data. Data are 60 sample and 100 gene, and there will be some numerical expression values. But you also have given labels. So there are two types of groups present. Some genes are tumor genes and some are normal genes. And then maybe, this data would have come from different time, from different labs, okay, from different batches. So batch effect is introduced. So for introducing batch effect that we'll introduce later on, first we need to define what a batch and what a batch 1 and what a batch 2, right? And then for that, half of the samples we are, giving it a batch 1, and we are labeling batch 1 and half as batch 2. Then we are generating base expression matrix. So we are generating random numbers, okay? And random numbers, so how many? N samples, so 60. 60 samples we are generating, and we are generating for 100 genes. So this will be 60 by 100 matrix, okay? And the whole data, so the random numbers that we are generating, they follow normal distribution and they are centered around 5. That means the mean of this data should be around 5. And there will be some variations in the data, so we are setting standard deviation as 1. So basic statistics. As a bioinformatician, you must know these things, okay? You must not only know, but you should understand the context. N row, so rows will be samples and columns will be genes, okay? Then we will assign the names of columns for the matrix which we have named expression matrix. So for this particular matrix names. So what we'll do, we will, all the gene we are naming it as gene. Okay, so first we have this prefix as gene, and from 1 to n genes. So n genes, we have set the number as 100. So we are having the column name as gene 1, 1 to n genes, that is 1 to 100, right? So gene 1, gene 2, gene 3, gene 4. So how gene will be named over here is like gene. Okay, the first one will be gene 1. Then 1 to n genes value is 100. So the second gene name will be gene 100. Okay, so sorry, gene 2. The third gene name will be gene 3, and so on till gene 100, right? If you are giving a space over here, so space will be generated over here too. So it's upon you. When you're working with it's a standard norm, and it's it's better not to have unnecessary spaces. Okay, I do not prefer it. You can work with them, but I don't prefer space. Now let us. So we have generated a mathematical table till now, and we are just naming it. It's a 60 by 100 matrix. Some random numbers centered around 5, standard deviation 1. That's it. No biology. It's totally math. We'll add biology now. So in the table expression matrix, we will group some of the gene into the tumor subgroup from 1 to 20. Okay. And okay, so so group. So first group is tumor, and the first to 20th gene, we are assigning it as tumor gene, and you see +3 at the end. That means whatever value, they are slightly upregulated by 3 levels. And then the tumor genes from 21st to 40th position, we are down regulating it. Okay, and because in real world some genes are upregulated and some genes are downregulated. Then we are also adding some latent signals. Okay. So what are these latent signals? Let me tell you. What happens here is like from the 41st to 60th gene, and this is a for loop. If you don't know for loop, if someone is new to these iterations, data structures, data types in any of the programming languages, so this is a for loop. So what it will do, it will iterate from 41st to 60th gene, okay, and some extra, the expression values, they are just correlating it, right? So this is some math over here. Right now, I'm not going to explain this particular math. We're moving forward. So basically, if you want to understand the biology, here we are just adding some co-expression value. Now we also need to add batch effect. Now this is not biology. Till now we are adding biology, tumor upregulation, downregulation, then co-expression. This we are adding, and now we add a technical bias. So I've already told you here we have introduced two batches. Half of the samples were from batch one, and half of the samples were from batch two. Um, Here from the expression matrix, we are choosing all the samples from the batch 2, and what we are doing, we're slightly upregulating that because of different environment, maybe different protocol. Okay? Even if you're following same protocol, you do not expect to get the same exact output every time, especially in wet lab. You cannot set seeds when you're doing wet lab experiments, right? So yeah, so to simulate, some difference will be there, not as uniform as what we are encoding over here, that all the second batch is 1.5, levels up. No, it is not there. So even if you're simulating this data set, it is not 100% mimicking the real data set, right? But we are trying to understand that what noises are there. And sometimes if you're not a good bioinformatician, if you do not know biology, you will not able to distinguish between what exactly is a noise or what exactly is the underlying biology. Okay, so excuse me. Now we have added the biology, we have defined what kind of noise we'll be having. Are there any other things? Yes, several different dimensions, right? But right now we're doing just this. Now just convert them into data frame. So gene data, so these are some commands as data frame. Now this expression matrix that you have made a simple table, a simple n by n matrix, it is converted into a particular data structure in R. You have data frames in Python as well, in many other languages. Yes, we have them. A robust, a table, just like a table, right? And then in that, there are two groups we can subset. So these here we are adding metadata. All the samples are there. Okay. So how you're grouping it? By assigning them the values of group. For this group, starting from small g, you can go here, you see, half of the samples belonging to tumor group. Okay, and half of the samples belonging to normal group, that is group. So you are labeling. Out of this 100 genes, 50 are normal and 50 are tumor. Gene data as batch. So this is another label you're giving. Half of the sample randomly, whether regardless of whether it is a normal sample or a tumor sample, some are coming from batch 1 and some are coming from batch 2. I hope now you understood this data set. Let us run and let us have a look on how it is looking. So to run, just select everything. I mean, whatever, which chunk you want to run, and you can click over here. Otherwise, you can press Control and Enter. You see what we have? We have the data for batch. We have the data for why we got this. Okay, I've run these lines also. I did not intend to run this, but let us see. So we have batch, we have gene, we have group. So batch one, and some of the samples are in batch one, some of the samples are in batch two. Some of the samples are in group one, some of the samples are in group one, and some of the samples are in group two. So group are tumor and normal. Okay. Then we have some latent signals. So the expression of other genes are uh, affecting the expression of, we had some default values of expression before, but after giving this latent signal, the expression has been changed for some genes. Because yes, If one gene is expressed, maybe the other gene might be downregulated or upregulated. Some are tumor suppressing genes, some are tumor, some are oncogenes, right? And not only suppressing an oncogenes, but also, in a regular pathway, this co-expression happens. So this is also biology. So n genes, 100 genes we have, n samples, 60 samples we are having. Okay. And yeah. So these are the expression values for tumor and normal, right? Let us look at the data. So head of, we will look at batch, how batch is looking. B batch. Yeah. The first gene is from batch 1, the second gene is from batch 2, the third gene is from batch 1. Okay. So what it has done is alternating batch 1, batch 2. You will not get this in your real data set. Okay. Let us see, after batch, let us see head of group. Head group. So what does head command does? The head command shows the first few lines of your table, okay, of your data frame. Not table, we'll say it is data frame. Okay, data frame is a robust table. I'll just show you how a data frame looks like, just like a table, right? So head of group, so it is tumor, tumor, tumor. So I can see only tumor, whereas normal. So we can also explore tail of group. Okay, so from the whole data set, we have subset, batch, and group, right? So we are for the group, we'll look at the tail. Okay, so this is tail, these are normal band. So yeah, we have both tumor and normal. Likewise, if you just look all together, so you have tumor group from batch 1 and batch 2, and normal groups from batch 1 and batch 2. And then we will see latent signal will just give you numbers. Okay, now let us have a look at the gene data. So this is the matrix. This is sample 1, sample 2, sample 3, sample 4, gene 1, gene 2, gene 3, like that. So here first 50 genes we are visualizing. You can see some values are in negative, some are near 0, some are 1, some are 2, some are even 4, you'll see. Yeah. Yep, that's it. I think I'll wind up with this video right now. Just understanding the data set, it takes a lot if you're a newbie. Try to grasp this thing that you, and this is a very tiny, tiny, tiny data set, right? You'll get a huge data set and you'll get much more complications in real data set, right? You have to have good communication with your wet lab people and you have to ask them under what conditions they have worked with, then they will uh, brief you about the protocols that they have used. And as a bioinformatician, you should be aware of what noises can you incur in your analysis, and do not think that this is noise, so it cannot be biology, and this is biology, and this is not noise. You should be very clear about it. And that is why, as a bioinformatician, know your biology first. Okay. So I guess something I've learned today in this video, and I'll wrap it up for now. And the next video, we will plot the histogram.